Okay, so the second part of this activity, so you've done the first part where you compared these, you made a Venn diagram, and then you made a cladogram, a very simple one, but you made a cladogram that showed kind of the relationships between those. And this was based on the characteristics up here. Okay, what we're going to do now is we're going to use a database and a text file of cytochrome C nucleotide sequences, and we're going to see how um, how the results you had up here compare to the the um, the genetic sequences down here. Okay, and we're going to use a cytochrome C, which is called COX1. So what I've done is I've used this website to look up all the cytochrome C DNA sequences. I've put them in a file here, so you can click here to download those files, and you're going to open it up. And it's going to look like this. So what this is this is the order of nucleotides A, T, C, G that you need to make cytochrome C in a kangaroo. And this is a sequence you need to make cytochrome C in a bullfrog. Now, cytochrome C is a very important protein that doesn't change much. And it doesn't, if it does change, it's very deadly to the organism. So it's a conserved species, conserved protein, and um, all living things, especially animals at this point, have cytochrome C. So it makes it a really good protein or a good DNA sequence to compare. Um, so here's what you're going to do. So once you have all of these, I want you just to hit Command A, and I want you to copy everything. And then you're going to go to um, the Clustal website. And when you go onto the website, it's going to have proteins here. You need to change that to DNA. And then I want you to cut and paste those in, okay? And make sure you have this little key there because that's going to tell you the differences, okay? So I've basically put all those sequences. So what we're going to do is we're going to compare the DNA sequences of the COX1 gene between these 7 organisms, okay? So then all I do is I hit submit. It'll take a couple of seconds. It's not super quick. The job's currently running. You wait, grab yourself a cup of coffee, um, talk to your friend, get up and stretch. Oh, here we go. Okay, so what it's done is it's lined up all the genes. And what it's kind of showing you here is the little star means that the nucleotides all line up there. So you notice here they're all kind of slightly different. So it goes through this whole thing. So then all you got to do is count the stars to see the difference. No, I'm kidding. This will do it all for you. So if you go to results summary and you click down here to the percent identity matrix, this tells you what percent of the DNA is similar. So this is kind of like a little matrix. So if it says lamprey here, it's lamprey up here. Rhesus monkey is a rhesus monkey here. This is human. This is tuna, bullfrog, kangaroo, snapping turtle. Okay? So notice lamprey and lamprey surprisingly have 100% similarities. But the lamprey and the rhesus monkey have 73.94% similarities. Okay? Um, so, obviously, if they're more closely related, they're probably more related to each other. So notice the rhesus monkey, which is the second row here. Notice how much it shares with humans. So that suggests out of all of these animals, we're more closely related to the rhesus monkey than we are to the humans are closely related to rhesus monkeys than any other organism there. Okay, so this is a pretty powerful way to kind of do some comparisons there. The other thing I want you to do is go up here and click on Phylogenetic Tree. So The phylogenetic tree is going to organize these very much like a cladogram, um, and based on the DNA similarities. Okay, so if you look here, you can see that it's put the lamprey and the tuna closer to each other. So there's a split there. So those are the two closest because they're exactly one node apart. Bullfrog, snapping turtle is 1, 2, 3 nodes apart, and so forth. Notice the kangaroo is kind of off by itself a little bit, right? It's a pretty unique animal. Um, so it doesn't have anybody really common here. But the rhesus monkey and the human, obviously we share a closer common ancestor than we do the kangaroo. But rhesus, human, and kangaroo are more closely related than the lamprey and the tuna. So this is how you can use a phylogenetic tree to help you with this. So what you would do is take a look at this. You may take a screenshot of it, put it in with your notes, and kind of see does that make sense compared to your very simple cladogram you made. Okay, so this is just kind of a practice of using that, because the next part, activity B, looks at hominid evolution. So here we're looking at humans and our ancestors. So hominins are us, Neanderthal, Homo habilis, some other organisms. And then our kind of our closest related ancestors, chimpanzees, gorillas, orangutans, gibbons, old world monkeys, new world monkeys, tassars, lemurs, and lorises. And what I've done for you is I've done all the COX-1 DNA sequences for those. So you can take those, you can download those, put them into Clustal Omega, and compare those sequences, come up with with a cladogram like this. And then I want you to kind of use that cladogram and this this table, and basically I want you to describe how well they fit. Okay, so using the DNA sequences, does your tree above fit with your data? Okay, so remember, if they're time-wise, the further back the ancestors are, the further back, the less relationships there are between those two. So I want you to kind of analyze that, and I want you to kind of have a little page ready to explain that. Okay. The lesson summary has some stuff about this information too. And then if you have time, I want you to pick one of these 3 articles and write a little bit about it and what it says about humans. And I want you to read one of those 3 and be able to talk about it. All right. So hopefully that will keep you busy. If you finish that, there is, if you go to review work, quizzes, and tests, there is some review work. Sorry, there will be some review work. There's a big classification sheet that you can start working on, and just to start revising for your test. And I'll have some practice questions for you next class. Okay? All right. Thank you.